Hello everyone, my name is Nurul Hakimah Khairani binti Ahmad. So today I will talk about DNA. DNA is also known as deoxyribonucleic acid. Inside the cell, we will most commonly find double-stranded DNA in which two strands that is intertwined to form a double helix. So firstly, James Watson and Francis Crick are known to be the first one who discovered about DNA, but then the truth is actually DNA was first discovered decades before by a Swiss chemist called John Friedrich Mischer. So here is a picture of unwound double helix. Here we can see the chemical structure inside clearly. Each strand is a polynucleotide, which means that the strand is made up of many individual units called nucleotides. So let's see what is inside the nucleotide. A nucleotide consists of three main components, which is firstly the 5-carbon sugar, a phosphate group, and one of 4 possible nitrogenous bases. So let's move to the pentose sugar. Pentose sugar in DNA is called deoxyribose. Pentose sugar contains 5 carbon atoms. If we go from there, we can see that there is a phosphate between the 5' carbon of one sugar and the 3' carbon of neighboring sugar. The sugar is called deoxyribose because it is missing a hydroxy group at the 2' carbon which is present in ribose. Phosphate is a chemical compound made of 1 phosphorus and 4 oxygen atoms. It is called as phosphate group when it is attached to a molecule containing carbon. It consists of phosphate ions and help bind the two deoxyribose molecules together. Here is a picture of nitrogenous bases. There are 4 main types of nitrogenous bases in DNA, which is adenine, guanine, thymine, and cytosine. A nitrogenous basis is simply a nitrogen-containing molecule that has the same chemical properties as a base. Of these nitrogenous bases, adenine and guanine are purines, which are aromatic compounds attached to an imidazole group, or known as double ring, while cytosine and thymine are pyrimidines, which are one-ring aromatic compound. Nitrogenous bases being hydrophobic, it tend to face inwards of the double helix, pointing away from aqueous environment. They are particularly important since they make the building blocks of DNA. The bond between the stacking of DNA are kept in close contact via van der Waals interactions. This also sometimes be called as stepladder model. The rings of the ladders are formed by complementary pairs of nitrogen bases. Adenine is always paired with thymine and guanine is always paired with cytosine. Nucleotides attach to each other in the DNA strand by phosphodiester bonds. The phosphate group of one nucleotide binds to the 3' oxygen of the neighboring nucleotide. We can see that the sugars and phosphate groups make up the DNA backbone. Actually, the carbon numbering is key to describing the directionality of DNA strand. 5' to 3' hooking with the sugars. There are intrinsic orientation difference between the two strands. On top of the strand, you can see that the 5' carbon of each sugar is on the left and the 3' carbon is on the right. The strands are also sometimes be called as Watson and Crick. It is because of in the early 1950s, American biologist Dean Watson and British physicist Francis Crick came up with their famous model of DNA double helix. Although the nucleotides come together through covalent bonds in the backbone, the two DNA strands interact through non-covalent hydrogen bonds between the bases. Each base forms multiple hydrogen bonds with its complementary base on the opposite strand. These two strands associate hydrogen bond between chemically complementary nitrogenous bases. As it bound together by hydrogen bond, each unit is called as a base pairs. There are 3 hydrogen bonds connecting between cytosine and guanine while two hydrogen bonds connecting adenine and thymine. The double helix structure of DNA is highly regular. Each turn of the helix measures approximately 10 base pairs. In addition to the hydrogen bonding between the bases, the stacking of the bases also stabilizes the double helix structure. So let's move to the function of DNA. DNA have a lot of function. The main role of DNA is that it is a long-term storage information. It is often compared to a blueprint since it contains the instruction to construct other components of cells such as proteins. The DNA segment that carry genetic information is called as genes, but other DNA sequences have structural purpose and involved in regulating expression of genetic info. In eukaryotes, such as animals and plants, DNA is stored inside the cell nucleus for most of the life cycle of the cell. But during time when the cell replicates, DNA is arranged in the form of structures which are known as chromosomes. For prokaryotes such as bacteria and archaea, DNA is located at the cytoplasm. So basically, the major function of DNA is to encode sequence of amino acid residues in proteins by using genetic codes. So, thank you for hearing. That's all from me. Thank you so much.